Good morning, students. Following double fertilization and endosperm development, now it's turn to study embryo development in an angiospermic plant. Zygote give rise to proembryo, globular embryo, heart-shaped embryo, and mature embryo. This process is known as embryogeny. Embryo develops at the micropylar end of the embryo sac where the zygote was situated. Most zygotes divide only after certain amount of endosperm is formed. This is the adaptation to provide nutrition to the developing embryo. The seeds differ greatly, but the early stages of embryogeny are similar in both monocot and dicot seeds. The zygote give rise to several structures like proembryo, globular embryo, heart-shaped embryo, and mature embryo. A typical dicot embryo consists of embryonal axis along with two cotyledons. The portion above the cotyledon is known as epicotyle and the portion below the cotyledon is known as hypocotyle. Epicotyle terminates in the plumule or shoot tip while hypocotyle terminates into radicle or root tip which is covered with a root cap in order to prevent the root tip while growing roots. Embryos of monocot possess only one cotyledon. In grass family it is known as scutellum which is situated laterally to the embryonal axis. At its lower end of the embryonal axis has root tip that is radicle and root cap enclosed in an undifferentiated sheath known as coleorhiza. But the portion above the axis is covered with a hollow foliar structure known as coleoptile. Coleoptile encloses shoot apex and a few leaf primordia. The major difference between the embryo of monocot and dicot seed is presence of one cotyledon in monocot and two in dicot. Now let us see the development of seed from the ovule. In angiosperms, the seed is the final product of the sexual reproduction. Hence, it is often described as a fertilized ovule. In angiospermic plants, seeds are formed inside the fruit. A typical seed consists of three main parts: seed coat, the outer covering, cotyledons, the reserved food is present over here, and the embryo axis, which will give rise to the baby plant. The cotyledons are simple structures which are generally thick and swollen because they have stored a large food reserve. On the basis of residual endosperm, seeds are of two types, albuminous and non-albuminous. Non-albuminous seeds are also known as X-albuminous seeds. They are non-endospermic, that is, no residual endosperm is found. So all the endosperm is consumed during embryo development. Some examples are pea, groundnut, etc. We can see in the diagram that no endosperm is seen, only cotyledon and the embryo is seen over here. The seeds which retain endosperm are known as endospermic seeds or albuminous seeds. Endosperm is not completely consumed during the development of the embryo. Some examples include wheat, maize, barley, castor, coconut, etc. We can see the endosperm in the diagram of the maize seed along with the cotyledon and embryo. Occasionally in some seeds such as black pepper and beet, remains of nucellus are also found. These remains are known as perisperm. Integuments of ovules get hardened to form seed coat while maturation of seed from the ovule. Micropyle remains as a pore in the seed coat which facilitates the entry of oxygen and water while seed germination. As and when the seed matures, the water content gets reduced and seed becomes relatively dry, that is, only 10 to 15% of moisture remains in the seed, due to which the general metabolic activities of the embryo slower down and embryo enter a state of inactivity known as dormancy. If favorable conditions are available, that is adequate moisture, oxygen, suitable temperatures, the seed germinate. A seed consists of a baby plant, that is embryo, which is when sown in the soil give rise to a complete plant in presence of sunlight, water and nutrients from the soil. As ovules mature into seeds, the ovary develops into a fruit. Let us see how fruit develops from a flower. When a flower opens, the reproductive parts are exposed to the pollinators. The pollinator transfers the pollen, leading to the fertilization of the ovule present inside the ovary of that flower. After fertilization, as the ovule converts into seeds, the same time ovary converts into a fruit. This completes the formation of fruit. The transformation of ovules into seeds and ovary into fruit proceed simultaneously. The wall of the ovary develops into the wall of the fruit known as pericarp. Fruits may be fleshy as in guava, orange, mango, or dry as in groundnut, mustard, etc. Many fruits have evolved mechanism for seed dispersal depending on the number of seeds produced by the plant. Mostly, while fruit develops, the other parts degenerate and falls off. But in few fruits, they remain as in apple, cashew, strawberry, thalamus contributes in the fruit formation. Such fruits are known as false fruits. However, the other, which develops only from the ovary, are known as true fruits. Although in most of the species fruits are results of fertilization, but there are few species in which fruits develop without fertilization. Such fruits are known as parthenocarpic fruits. Banana is one such example. Parthenocarpy can be induced through the application of growth hormones, and the fruits such formed are often seedless. We can see true and false fruits. Example of true fruit is a grape. Seed is inside and the ovary is transformed into fruit. While in apple, that is a false fruit, we can see the seeds along with the fleshy part made up of thalamus. The fleshy part is outside the pericarp because the flower parts are fused and swollen to form the edible food. While in grape, pericarp is the outermost covering. Parthenocarpic fruits like banana. Now parthenocarpy can be induced in custard apple, tomato and several other fruits in order to obtain seedless fruits, which is very favorable and in demand. In the next lecture, we will be covering the remaining topics of this chapter, that is advantage of seeds produced in angiospermic plant, apomixis and polyembryony. I hope you have understood all the events taking place in the sexual reproduction in a flowering plant.